Next, I'd like to introduce Anandani Nellan. Dr. Nellan did her pediatric training at the University of Virginia, and then did her pediatric hematology oncology fellowship at the Joint Hopkins NCI program, and then went out to Colorado to do a neuro-oncology fellowship with our friends Nick Foreman and Rajiv Vibhakar. And then she returned to the NCI, where she's been working in CAR T-cell therapy for ependymomas. And so she's going to speak to us about CCL2 mediated immunosuppression in CAR T-cell function in posterior fossa ependymomas.
Thank you for the opportunity to speak today. All right, so I'll be talking to you today about a project that spanned over the last 3.5 years in studying CAR T cells in posterior fossa ependymoma. So first, some overview. So ependymomas or ependymomal tumors can occur across age groups. They can occur in the supratentorial space, so higher up in the brain, in the posterior fossa, so that's lower region of the brain, so brainstem, fourth ventricle. It can also occur in the spinal cord. The vast majority of ependymoma that affects children occurs in the posterior fossa, and it's this tumor type that I study in the lab. And so what this figure is trying to show you, so first we start off with what's a CAR T cell. So CAR T cell is a modified T cell, typically a patient's own T cell, where we insert a gene. And there's multiple methods of inserting the gene. A common method is using a virus that can insert the gene. And what does the gene encode? So the gene encodes a protein that recognizes tumor cells, so typically a protein that comes from an antibody. So this is what gives the CAR T cell specificity to recognize a particular target. And then it also incorporates signaling regions from a T cell. So then now you have a all-in-one therapy that can recognize a tumor and then also mediate tumor cell death. Then moving from left to right, you see a brain with the ventricle system, so that's the cerebrospinal fluid within the brain. And then in the fourth ventricle is a posterior fossa tumor. And the picture below shows you that anytime you develop a therapy to target tumor cells, it's never the tumor in isolation, and you have multiple other cells that are kind of interwoven with your tumor. So you have myeloid cells, stromal cells, endothelial cells, you have the extracellular matrix, and then with the brain, you also have normal neurons and glial cells. And then next you see how patients with brain tumors are receiving CAR T cells in the clinic. So one method that's being evaluated is instilling CAR T cells directly into the ventricular space. So the idea here is that the CAR T cells are a little closer to the tumor itself that you're trying to treat, and perhaps you're reducing toxicity or T cells all over the body and just in the CNS region. So that's one method that's being employed. There are other studies looking at delivering CAR T cells to the peripheral blood. And both methods are being used for patients with ependymoma, and so targets that are in the clinic are HER2 as well as B7H3. So target I've been studying is HER2. So the HER2 is widely expressed in ependymoma, so in most posterior fossa tumors have elevated levels of the ERBB2 gene, and compared to normal brain. And the construct that I evaluate is from the Herceptin, or the trastuzumab monoclonal antibody. So the binding region recognizes HER2, and the signaling domains incorporate the CD8 alpha transmembrane domain with the 4-1BB co-stim. So what you see up top is the antibody portion of a CAR, and what you see at the bottom are the signaling regions that come from a T cell. And the two models we study in the lab are two patient-derived xenografts, and then that can also be cultured as cell lines. So these are two patients that had a metastatic relapse of posterior fossa ependymoma, and the tumors were cultured in the lab, implanted in mice to be able to propagate it and then to further study. And ependymomas have been notoriously difficult to study in the lab because it's difficult to grow on plastic, sometimes even difficult to grow in mice. And so these are two, so it's 811 and 928, and both cells express high levels of HER2 on the surface. And what you see here is co-incubation of tumor cells with CAR T cells. And what you see is that you have high levels of cytokines, so TNF-alpha, IL-2, interferon gamma, when you co-incubate HER2 CAR T cells with HER2-positive tumor cells. So 811 and 928 have high amounts of cytokine that's produced by CAR recognizing the tumor. And as controls, we have NALM-6 leukemia cells that are HER2 negative, but CD19 positive. And so we have an additional control of CD19 CAR T cells that are used in subsequent experiments that you will see. So CD19 CAR T cells would be what we would call an irrelevant CAR in these experiments. So it's another CAR T cell, but it doesn't actually recognize ependymomas. And here, what you see is cytotoxicity or killing of tumor cells by CAR T cells. And you can see that we get some very nice killing of both 811 and 928 ependymoma cells when you co-incubate it with HER2 CAR T cells. And that doesn't happen with the antibody itself alone or with CD19 CAR. And so now the model system. So to model this disease in mice, we use very immunodeficient mice, so NSG mice. These mice lack B and T cells. They have non-functional NK cells, have very defective dendritic cells and macrophages, but these mice still do have neutrophils and monocytes, which make up a large portion of its immune system, and this becomes relevant later. And so how do we instill the tumor? So you use this device. Mouse is anesthetized. It can't move. Shouldn't be able to feel what you're doing. So the mouse is immobilized in that device, and then a needle instills 3 microliters of tumor cells directly into the fourth ventricle. And the latency is long, so it can take up to 2 to 4 months for these tumors to form. So it's definitely not a speedy model. But it has allowed many preclinical studies since it was developed. And here is a schema of an experiment. So you have mice that were implanted with tumor, and we have 2 treatment groups. So 5 mice getting the CAR that recognizes tumor and 5 mice getting irrelevant CAR. And we used a tail vein injection method to treat mice. And what you see here are MRI images of mouse brain, and you can see in blue delineates the tumor in the mice that received HER2 CAR IV, and red are the mice that received the CD19 CAR IV. And what we saw was right around 2 weeks, mice get very sick after getting HER2 CAR IV, sicker than even the control group. And what we learned was that these mice have pretty massive expansion of their tumor after receiving CAR T cells, and the mice that can tolerate that expansion do end up having some tumor regression afterwards. And then now we'll look more closely at the mice at tumors right around that 2-week mark, both after getting HER2 CAR IV or CD19 CAR. So this is a growth curve where you see increase in size of the tumor and then decrease in the mice that tolerated it, and then when the mice that got CD19 CAR, the tumors just keep growing. And one mechanism of CAR T cells not working is loss of the target. So when you look at these tumors, there really is no difference when looking for HER2 expression on the surface at the early time points or at the late time points. And then we submitted our samples for single-cell RNA sequencing. And so what you see here is a total of a little over 6,000 tumor cells. So it's all the same tumor. So it's this 811 patient-derived xenograft model. And what we have here are HER2 CAR-treated intracranial tumors, 19 CAR-treated intracranial tumors, and as controls, we have untreated intracranial tumor as well as a subcutaneously grown tumor. So it's all the same tumor, just slightly different conditions. And what you see at this UMAP plot is that the untreated tumors seem to cluster together on the left, so the yellow and the orange, and the tumor cells after CAR treatment are intermixed and cluster to the right, so the blue and red. So each dot is one cell. And in terms of trying to understand what we were looking at, so I used a gene signature that was published in this important paper that came out of the Foreman lab at the University of Colorado, looking at where they performed single-cell RNA sequencing of 26 patients and with ependymoma. And what they really discovered is, yes, there's absolutely this heterogeneity in tumor samples, and there are various subpopulations. And some of the key subpopulations in posterior fossa ependymoma are these ciliated and transportive cells, which are much more differentiated. Ciliated cells made up the majority of cells found in patients' tumors, and have a lot of common genes with normal ependymal cells in the brain. And then you also have these smaller subpopulations of cells looking like they are undergoing epithelial to mesenchymal transition. So you have this undifferentiated population and a mesenchymal population. So the undifferentiated population have very high levels of FOS and JUN, sort of the hallmark genes in that category. And the mesenchymal population has very high levels of NF-κB pathway-related genes and chemokine genes, so CCL2, CXCL1, CXCL8. And in terms of how is this clinically relevant, the patients that had more undifferentiated mesenchymal cells had a more aggressive clinical course and worse outcome. What we see here is that there seems to be a change in terms of the signature of genes in MAF811 tumors after treatment with CAR. And so you have less of the ciliated and transportive gene signature, and you have more of the undifferentiated and mesenchymal gene signature in the HER2 treated and the 19 treated animals. And I think, some reasons why. So when you give HER2 activated human T cells to an immunocompromised mouse, you're going to get massive expansion of T cells. There's lots of inflammation, and whether perhaps this response could be due to the inflammation that we're causing. And then next, looking at histology of the tumors, we see a very cellular tumor. And what you're looking at there is a stain for mouse myeloid cells. So there's tons of mouse myeloid cells, and there's a ton of human T cells that are found within the tumor. And this is a HER2 CAR-treated section. And with flow analysis, there are more monocytes that are found post-HER2 CAR treatment compared to 19 CAR treatment. Let me just go back. Okay, so there were a couple hundred precious myeloid cells that we were able to actually analyze in that sample. Many of those cells didn't actually pass quality control, that we couldn't use the data. But in the 226 cells that we could isolate from these same samples, so it's these MAF811 tumors in mice that were either intracranial treated with CD19 CAR, HER2 CAR, or untreated intracranial and subcutaneous tumors. What you find are in the myeloid cells, you have resident macrophages, monocytes, as well as granulocytes or neutrophils. So on the right, you're looking at a heat map where each column is a cell, and each row is a particular gene. And so the monocytes are predominantly found after HER2 CAR treatment, and CCR2 is a top gene that shows up in that monocyte population. So next, we wanted to look at what might be driving the myeloid cell infiltration post-treatment with CAR T cell in our model. And we looked at a panel of chemokines that could mediate myeloid cell infiltration. It was cross-reactive in mice, and CCL2 was one that really showed up. So there is a large amount of CCL2 that's made by 811 and 928 ependymoma cells at rest, and much more after co-culturing with HER2 CAR T cells. And we used a medulloblastoma comparison and a leukemia comparison model where you really don't see that same pattern. And interestingly, when we looked at our patient's CSF sample, so this is looking at spinal fluid at the time of diagnosis, so this is what the surgeon collected at the time of surgery, there were high levels of CCL2 that we could find in patients with posterior fossa ependymoma compared to a large number of normal controls. So the values for the normal controls came from another study of patients without tumor. And we did a larger experiment, so the same model, HER2 CAR versus 19 CAR, more animals. And when we did flow cytometry analysis, you can see high amounts of monocytes post HER2 CAR, and many of those monocytes express CCR2 on the surface. And we evaluated our, instilled the PDX into mice and treated with CAR in this NSG mice that has this additional mutation that makes it very monocyte deficient. So it's this extra KIT mutation. And so these mice are the predominant cell you can identify in the bone marrow and the peripheral blood are neutrophils. And so you don't find monocytes. And interestingly, it took an extremely long time for tumors to grow. So it's not a perfect comparison at all. The latency extended to beyond 6 months to even see tumors, and the tumors are smaller. But what we did see is that the CAR therapy does seem to work better in this model. And then next we evaluated a CCR2 inhibitor, and that does penetrate the blood-brain barrier. It's delivered subcutaneously. And so we have 3 groups here. So CCR2 inhibitors, that's TAK-779, compared to CAR alone or combination. And what you see here, so far left, you see the inhibitor alone, and those tumors are outlined in purple, and you can see those tumors get very large. And the mice have hydrocephalus, so you can see there is a backup of spinal fluid, and that's what you're seeing right above the large tumor that's outlined. And followed by the group that received CAR alone. And so you do have some activity, and the tumors do get smaller. So they first expand, then they get smaller. So those are the tumors outlined in blue. And then in the combination group, it's not perfect, but we do see some dramatic responses. So two animals with really just a line, maybe just some residual line of where the tumor was. And even in a mouse that had a very bulky tumor, we saw regression and some necrosis in the center. And so this is 2 just growth curves from the 2 different models, where it does seem like combining CCR2 inhibition along with CAR T cells does seem to increase its efficacy in this model. So what do I think is going on, and kind of what are our ongoing efforts in the lab to better understand this? So, we know that ependymomas can have baseline aberrant NF-kappa B signaling. And this is in part can explain why we see so much, the tumor cells can secrete a lot of chemokines at baseline. So we give CAR T cells. CAR T cells make a lot of inflammatory cytokines, one of them being TNF-alpha. And TNF-alpha can bind its receptor, which is pretty ubiquitous on many cell types. It's also on the tumor cell. So TNF-alpha binding its receptor can further drive down NF-kappa B activation in the tumor. And this in itself can actually lead to increased CCL2 gene transcription and then release of a lot of protein or chemokine. And this can attract monocytes that express the CCR2 receptor. And myeloid cells have been shown to inhibit T cell function, and I think in this context that the therapy we're giving is further driving this inflammation in the tumor and attracting myeloid cells to the tumor microenvironment that perhaps aren't necessarily helping T cell function. And I think the other piece is the fact that the treatment itself seems to have a shift in the gene expression of the cells, so that's a further area of study. And so I think in summary, So monocytes significantly infiltrate the brain after HER2 CAR T treatment and can suppress T cell function. Findings in the tumor within the brain after HER2 CAR treatment implicate the role of the CCR2, CCL2 chemokine pathway. Aberrant NF-kappa B signaling in PFA ependymoma tumors can drive secretion of CCL2 and then recruitment of CCR2 positive monocytes. And I really think that understanding tumor-mediated immunosuppression will be important in actually designing immunotherapy trials that will be effective for our patients. And so this work started in Colorado, so an acknowledgement to all of the people I worked with at the University of Colorado, my current lab members, and then my current funding sources through the Physician Scientist Early Investigator Program, the NIH Distinguished Scholar Program, and then previous funding sources at the University of Colorado, without which none of the work would be possible. So thank you.
Any questions? So the whole idea of immune cells being able to move in and target brain tumors is, for those of us who were raised in the the blood-brain tumor era and the blood-brain barrier tumor era where nothing gets through kind of thing. Have people looked at mechanisms for how these cells move through and get into the brain tumors? And is there anything about how they get in that would be either helpful or harmful in therapeutically thinking about these cells?
Yeah, absolutely. So yes, I think T cells, especially activated T cells, in the setting of a lot of inflammation in a patient, large numbers of T cells can enter the spinal fluid, can enter the brain. And we've seen that especially in trials of CAR T cells in patients with leukemia and other hematological malignancies. And then I think in terms of the mechanism, I think the nicest work comes out of people who study multiple sclerosis or autoimmune encephalitis. And yes, in the setting of inflammation, there are changes that occurs where cells that didn't have permission to enter now have permission to enter, or more permission to enter, with changes in adhesion molecules and other factors that just now permit a lot more cells to be able to enter spaces that it wouldn't in, say, steady state or a very healthy person.
Other questions? Okay, I have one more. So when people are thinking about human trials for these, and the mice clearly got into trouble from inflammation and the fact that the cranium has limited capacity for handling swelling, are people thinking about ways of prophylaxing that in people, either by putting in prophylactic shunts or finding ways to make sure that the pressure itself doesn't kill the patient?
Yeah, absolutely. So I think, yes, especially for the local delivery of CAR T cell studies, all patients are required to have an implanted catheter. And it's sort of a pop-up valve. So, you can relieve pressure that way, you can take off spinal fluid that way. Steroids, so just like a very, blunt way of trying to suppress inflammation to try to decrease swelling in the brain is used. And I think the other thing is that, these mice have really bulky tumors. Many already have hydrocephalus when we give them CAR. I think for a patient to be eligible, that would not be a patient that would be a good candidate. It'd be much better to undergo surgery first to make the therapy much safer.
Great. All right. Thank you.